all eukaryotes have two other RNA polymerases, that is polymerase 1 and polymerase 3, in addition to polymerase 2. These enzymes are related to polymerase 2 and even share several subunits, but they initiate transcription from distinct promoters and transcribe distinct genes. So, besides many similarities, they have different promoters and they transcribe different genes. Those genes encode specialized RNAs rather than proteins. Each of these enzymes also work with its own unique set of journal transcription factors. So these enzymes have their own journal transcription factors. TBP, however, is universal. It is involved in initiating transcription in all the polymerase 1, polymerase 2 and polymerase 3. Although TBP is the only journal transcription factor that is used by polymerase 1, polymerase 2 and polymerase 3, it has emerged recently that some of the other GTFs in fact have structurally and functionally equivalent components in the other systems. So some other GTFs are also used which have similar structure and functional units. Thus, for TF2F seems to have a counterpart in two subunits within polymerase 1, that is A49 by 34.5, and also in polymerase 2, that is C37 by 53. Likewise, TF2E like subunits are found in polymerase 1 and polymerase 3 enzymes. In addition, both these other systems include additional factors comparable to TF2B. The TAF1B factor in the polymerase 1 system and the BRF1 subunit of TF3B in the case of polymerase 3. Polymerase 1 is required for the expression of only one gene, that encoding the rRNA precursor. So, for the synthesis of rRNA, the gene which is responsible, that is transcribed by pol 1. There are many copies of that gene in each cell, and indeed it is expressed at far higher levels than any other gene. So perhaps explaining why it has its own dedicated polymerase. So as it has many copies and it is expressed at very high rate, that's why it has its own dedicated polymerase that will only transcribe this particular gene. The promoter for the rRNA gene comprises two parts. So it also has its specific promoter, and this promoter has two parts, the core element and the UCE, upstream control element. So there are two parts of the promoter. One is core element, the other one is UCE element.